So I'm also from the transplant program here at Columbia University, and this is my first time presenting in the myeloma crowd, and I think this is absolutely great. I was only vaguely aware of this program so far, and just going into the websites and the types of blogs, radio programs, and these types of roundtables, I think it's absolutely phenomenal, and the people and the type of content that's presented is absolutely outstanding. So I'll try to help as well, and I think I'm complementary to what Dr. Mapera was talking about, because ultimately the idea of cell therapy emerged from the field of transplant by taking cells from the patient and then transplanting them back, but adding another layer of innovation to it, which is to modify these cells, to force them to fight the cancer better than your own cells just are naturally occurring. So what you see here on this slide is actually a microscopic depiction of a T cell attacking a myeloma cell. I'm not sure it's actually a myeloma cell, but it could be a different tumor. But the idea is that we know that our lymphocytes in the body, and the T cells in particular, which are a type of lymphocyte, are capable of killing tumor cells. And the idea is that many patients with cancer, it just doesn't work correctly or doesn't work potently enough. And the question is, how can we harness these T cells, and how can we force them to kill cancer better? So this is where CAR T therapy came from. And I'm just going to give a brief summary, and then we're going to talk about some of these points one by one. So the name CAR T therapy is the name given to chimeric antigen receptor. This is what CAR stands for. And these are T cells that were taken from the patient usually and genetically modified in the lab, or another terminology used is genetically engineered. And then basically that designs them to recognize tumor cells better and destroy them in a more potent manner. They are a living drug, since these are living T cells. So you modify them in the lab, you give them back to a patient, and they continue to survive in the patient, being able to multiply. Their progeny looks exactly as the parent cells, and they can even persist for very long periods of time, with nowadays people who are 5 or 6 years out from a single infusion of CAR T cells that can still have detectable CAR T cells in their blood. So this persistence might be very critical to the success of this therapy. They're generally created from the patient's own blood, although when you start looking at this in various online resources, you'll see that there are also attempts to create off-the-shelf CARs, meaning that they're going to come from random donors, similar to a regular blood bank. They're going to be engineered ahead of time, and then you don't need to do this manufacturing process. It's just going to be sitting on the shelf ready for use. These are in more early phases of development, so I'm not going to talk about them. They're not yet being developed in myeloma. I feel, and I think many people in the field feel, that this is the most advanced form of immunotherapy, which we know over the past decade has a huge capacity of harnessing the patient's own immune system to fight cancer instead of using drugs, chemotherapy, and other modalities. So I think this is one of the most efficient ways that we may have in the future to do this. The history is actually quite short. Well, this doesn't present all of the history, because to know how to genetically engineer cells was already found in the late 80s, actually at the Weizmann Institute, which one of our participants here mentioned to me earlier. So the history of this, I was fortunate enough to be at the University of Pennsylvania when my colleagues treated the first 3 patients with chronic lymphocytic leukemia, another hematological cancer. And all 3 of those patients had a response, where 2 of them had a complete response, which is sustainable until today. They were treated in 2010, so they're 8 years after a single infusion of CAR T cells. And these were patients who've had 7 lines of therapy for CLL, had high-risk disease, and are basically, as we could tell today, cured of their disease. This is a picture from the FDA panel that discussed this product. And this is Emily Whitehead, which became pretty famous in various New York Times articles and other places as being the first pediatric acute leukemia patient who was treated, and in fact suffered significant side effects. And on her, basically, people have figured out how to treat those side effects. And she is without leukemia and alive, I think now 6 years out of therapy, going into high school. So these were kind of a few of the miraculous first stories about this. What is the rationale for targeted cell therapy? So as I mentioned earlier, the T cells, which are basically a type of lymphocytes, we know that they're capable of killing cancer. They're capable of killing any type of abnormal cell that occurs in our body, the same way they're capable of killing viruses and bacteria and any other invaders that are unwelcomed in our body. The issue is that we need to redirect them against tumor cells to make them more effective, because we know that people get cancer all the time, and part of that is because the T cells are just not capable enough to eradicate cancer early enough or when it's already established. So what happens if you redirect them to recognize tumor cells? This will overcome many, many limitations that our current agents might have. So the advantages of this approach is, first of all, the specificity, because when we engineer those CAR T cells, we force them to recognize only a single type of protein that's expressed only on a single type of cells. And we use an antibody, which is the best way, or this is our best recognition mechanism for this. They're very potent, and they're what we call serial killers. And that we saw even in the first CLL patients who were treated. These were patients with very large lymph nodes, large numbers of CLL cells in the blood, and they only got a few millions of T cells. And all the disease was eradicated within a few weeks. So what we realized there is that one CAR T cells can destroy one tumor cell and then move on to the next tumor cell and destroy that one as well. And someone did the math that one CAR T cells could basically destroy thousands of tumor cells just going one after the other. There's an amplification here because those T cells are, as I mentioned, living drugs. So they go back into the patient, they belong there, they're still alive, they would continue to do whatever T cells do, which is to multiply in the patient, and their progeny are exactly the same. So you get an amplified response, and very importantly, or at least some people think it's important, that this creates memory. So when you have these long-term, long-living T cells in the body, this starts to look basically like a vaccine approach. This is like you get a vaccine for measles when you're a kid, and you're protected against measles for the rest of your life. That protection is given to us by T cells. So basically these T cells might be able to basically vaccinate the body against the cancer. So if the cancer is making any attempt to come back, then there's already going to be an active process to prevent that from happening. Well, what do the CARs really look like? And I don't want to go into the technical details of the genetic engineering, but imagine that there is a gigantic T cell here, and this is just a zoom in on its surface. This is the surface of the T cell. So the CAR construct is basically this thing. It's a completely genetically engineered molecule that has an outside extracellular portion, which is basically an antibody. This antibody knows to recognize something that's presented on the tumor cell. And it's connected from the inside of the cell to a signaling domain that activates the T cell, right? It's not enough that you would recognize the tumor. You should tell the T cell, okay, now you start destroying stuff. So all of this killing, growth, survival, the multiplication of these T cells is triggered by this very simple construct. How does this really happen? And this is, bear with me with the graphics, because this is the first time I'm using this slide. So we have a myeloma cell. The myeloma cell expresses all kinds of proteins. One of these proteins that is very commonly expressed in myeloma cells is called BCMA. That is the target that is currently looked at for CAR T cells. This is the T cell from the same patient. T cells have receptor already to recognize all kinds of targets, but our T cells have these recognition mechanisms against, again, viruses, bacteria, fungi, all kinds of other things. In order to force them to recognize BCMA, we use a viral vector, which is very similar to all kinds of naturally occurring viruses, such as even the HIV virus. But they're neutralized of any potential bad effects, so they can't cause disease. But we put them in the cells, and this is done in the test tube in the lab. And it generates or integrates DNA into that cell that forces the cell to express the CAR. So at this point, this T cell doesn't only have its original native receptor to recognize viruses and things like that. It now has a unique receptor, which is the CAR basically, that recognizes specifically BCMA. You give these T cells back to the patient. These T cells find the target, the T cell gets activated, the myeloma cell dies. And the cool thing, as I mentioned earlier, this CAR T cell, nothing happens to it. It's not exhausted. It doesn't get tired in this process. It just moves on to the next myeloma cell. So this is really kind of the premise behind this type of therapy, which I think was considered 10 years ago science fiction. So what does the patient journey look? This all sounds like it's a lot of lab work, viral vectors, genetic engineering. What does this really look for a patient who wants to get this type of therapy? So this is the best one I could find, short of drawing one myself, which would never work. This is a patient that for whatever reason is naked here, but take into account that you can undergo this procedure fully dressed. The first step is to take T cells from the patient, and that is done because there's the starting material for this manufacturing process, for lack of a better word, requires quite a good number of lymphocytes. So that is not just done by a blood draw, it's done by a leukapheresis procedure. Anyone who's familiar with autologous transplants or who had one knows this process very well. It's even simpler than the apheresis we do on autologous transplants because this is what we call steady state apheresis. There's no need for growth factors. There's no need for chemotherapy prior to that. You just walk in the morning, get connected to an apheresis machine, get your lymphocytes collected in a bag and go home. These bags go to a manufacturing facility that, if we're talking about commercial products, they're in factories, basically, that were established for this purpose. They undergo the manufacturing process that I just described in the previous slide. They get a virus that activates this CAR on their surface. They get expanded, so we'll get millions and millions of them. And ultimately, they're shipped back to our center in a process that takes somewhere between 14 and 21 days. At this point, the patient basically has not received any form of specific therapy, so they go home, and if they're on some form of therapy to moderate the disease progression, they can continue with that. And then they come back to us to get the cells administered, and in order to create what we call a niche, some space for the cells to expand in the patient, we do give some chemotherapy, but this is a much milder chemotherapy. That's what's used in transplant or in other forms of aggressive cancers. It's fairly on the milder side, and we even do it as an outpatient. And then you get the cells infused. This is a one-time infusion and a one-time treatment. There has been little experience now with recurring infusions, and it's not clear that they're even necessary. This is what the bag looks like. So this is what you're going to get, and this is just one example from one of the manufacturer, which is already a commercial product called Kymriah. It is not for myeloma because it does not target BCMA. It targets a different type of antigen, but this whole thing fits in the palm of my hand. And this thing contains millions and millions of cells, and if you can see what's unique to this compared to other medications that come from companies, this has patient's name on it. So we give the company information who this is coming from, mostly to make sure that this comes back to the same patient, that there's no mix-up along the way. So these are patient-specific cells. So how well does this work? To give you a little bit of background, since this has been really in development now for 7 or 8 years, most of the experience so far has been in aggressive lymphomas and in acute leukemia in children, or acute lymphoblastic leukemia specifically in children. And the target for these CAR T cells has so far been CD19, which is something that is not heavily expressed on myeloma cells, so we can't really use it there. There's been a number of clinical trials that have either been completed or are about to be completed. And what's phenomenal about this, so these are all patients who have been refractory to multiple lines of therapy. You couldn't go on these studies if you haven't failed or progressed after a minimum of 2 lines of therapy, and many of those patients have had far more than that. And the response rates are still in the range of 82%. And what's more critical is that the complete remission rates are more than 50%. So more than 50% in these diseases have went into a complete response. In ALL, it's even more dramatic. There was up to a 93% response described in one of those studies. And I know you're already familiar with the terminology of minimal residual disease. almost all of these responses were MRD negative, so which means complete eradication of the tumor, including in patients who've had massive amounts of disease. And I think what's even more attractive, or maybe that is the main point, the main takeaway message from this, is how durable this is. And in lymphoma, it's been shown that beyond 1 year after a single infusion, 40% of patients are still in a complete remission. And there are now patients beyond 1 year, and there have in fact not been any progressions beyond 1 year in the lymphoma study, 60% in the acute leukemia study. We don't have much follow-up beyond that, but I've shown you Emily Whitehead, who is 6 years out of therapy with no evidence of disease. Based on these phenomenal results, both of these products have been FDA approved. So what's happening in myeloma? So in myeloma, we need to pick a different target because CD19 is not expressed or not expressed enough on myeloma cells. And the most used target so far by most trials is a protein called BCMA. And I'm just going to show you what I think is the most exciting data that was shown at the American Society of Hematology in December for a CAR T cell called BB2121, which is a CAR T cell. Its structure is exactly the same as what I've shown you earlier. It's just that the antibody on its surface doesn't recognize CD19, it recognizes BCMA. That's really the only difference. This is from a phase 1 study that treated 21 patients. You can see the age range here. 37 is definitely on the young side for myeloma, but up to age 74 have been enrolled on this study. Almost half of them had very high-risk cytogenetics. I think the median prior lines of therapy was 7. So these are patients who have really failed and have tried to go through multiple types of therapies, have probably been already enrolled on other clinical trials. The most treatment experienced patient have had 14 lines of therapy on this study. And everyone was transplant experienced, but that's not mandatory to be included. Everyone had Velcade and Revlimid. Most of the patients have had all of the series of the existing approved therapies. And this is what the outcome of this trial was. 94% of the patients responded. 56% had a complete response or a stringent complete response. Another 33% had a very good partial response, a VGPR, which we know that in patients who have been priorly treated with so many lines of therapy is quite a challenge to see even at these rates. And another 6% of partial responses. And by 6 months, 80% of these patients were still without progression and have not passed away from the disease. I'm just showing you the results of one program. I would mention, not to burden you with too much data, that there were at the same conference at least 2 additional BCMA CAR programs that were presented with similar, slightly different numbers, but overall the results of this are quite promising. This is a complicated slide, so I'm not going to review this in detail, but when we present phase 1 studies, because they usually include a small number of patients, we frequently present patients one by one, just to show how long each patient was maintained on therapy or following therapy and what was their best outcome. And a few things I would point out here. So time 0 is the time of the infusion. Remember again, I'm going to say it again, none of these patients got any subsequent therapy. This was a single infusion. We have a few patients who have been followed for more than a year, and they have an ongoing complete response. This light green one is a complete response. There have been a number of slowly evolving responses. So patients, for example, this one who initially had a partial response, then had a complete response, and then had a stringent complete response 58 weeks after the treatment. So these cells continue to work in vivo. They continue to work in the patient. If they didn't take all the disease away initially, their work continues and persists even after that. So even if you have a partial response, that does not mean at the end of the day that you wouldn't have a complete response at a later time point. The red stars are patients who've experienced MRD negativity. I think it's the red star, right? Yeah.
These are different dosages?
Yeah, so there are different dosages here. So you can see that the first dose, the first 3 patients had short-lived responses and progressed, which is why they went up to a higher dose, which is where you start seeing the more relevant responses. And the doses that they're taking to the next clinical trial are in this range of between 150 and 450. So MRD negativity, I mean, just look at the number of red stars here on the slide, and you could see that MRD negativity in those heavily pretreated patients was quite frequent. Keep in mind, these are only 18 patients. So we need more numbers, we need greater numbers, we need these trials to expand, and we need to open them here at Columbia to get our patients into them. And then hopefully this will become a reality for as a standard of care. This is what this particular company is doing as a follow-up study. They're calling it the Karmis Study, which I think is a really cool name. And eligible for this trial are the relapsed and refractory patients who've had at least 3 prior therapies. We may have additional trials that would be appropriate even for patients at an earlier phase. They undergo the same process I've just described to you. It's an apheresis at steady state, manufacturing, infusion with a little bit of chemotherapy, fludarabine and cyclophosphamide prior to the infusion. So just to show you the flip side just a little bit about what the side effects might be of this therapy. We don't see significant infusion reactions because these are the patient's own cells. So there's no reason why there would be a significant infusion reaction. Tumor lysis is something that is rarely seen, so I'm not really going to talk about it. Cytokine release syndrome is an important and common side effect, which is one of the reasons these treatments occur either in the hospital or that we ask the patients to stay in the close vicinity of the hospital so they can reach us quite quickly. And there are neurological side effects that include either changes in mental status, confusion, sleepiness. Some of them can be very scary to the patients and their family members. They have been shown to be reversible in almost universally. In almost all patients, these were completely reversible. And there's some drop in blood counts because patients do get chemotherapy prior to this. Just a little bit more information, and these will be a couple of my last slides. Cytokine release syndrome, as I mentioned, is probably the most common and most scary side effect. It results from the release of cytokines as these T cells find tumor, kill tumor, and multiply in the patient. They release a lot of inflammatory mediators that we call cytokines. This could manifest as high fevers, chills, lowering of blood pressure, lowering of oxygen. Occasional patients will have to go to the intensive care unit to be monitored and treated there. And there have been treatment management algorithms that were developed. And we know that blocking a certain inflammatory cytokine called IL-6 has been quite efficient in reversing this process. And this is one example of a patient who starts to have cytokine release syndrome. This is the temperature curve. Temperatures that could go up as high as 105.3. That at this point gets Tylenol and cooling blankets and antibiotics, and at this point starts getting a little bit too sick with maybe lowering of blood pressure or starts requiring oxygen, gets a dose of tocilizumab, which is a drug that blocks IL-6, and as a result of that, there is almost immediate drop in temperature and the patient recovers. And in patients who don't respond well to tocilizumab, we can always use steroids as well to kind of tone down the response of the cells. This is when this happens. This is most of the action happens really within the first 14 days. These are not long-term or delayed side effects. Cytokine release syndrome starts occurring either in the first day or within the first 7 days. Neurologic side effects may linger a little bit longer. But by day 28, in most patients, most side effects have resolved. And we ask patients to not operate heavy machinery up to 8 weeks, but other than that, they should be fine. Just to wrap up and show you what else is going on with CAR T cells, these are just a slide that shows you how many ongoing clinical trials there are with CARs against these multiple targets. 56 trials in B-cell lymphoma, 67 trials in non-Hodgkin lymphoma, 90 in leukemia. And since this seems to be so exciting on the hematologic malignancy side, our solid tumor counterparts have started incorporating them into their treatment paradigm, and there are clinical trials in a variety of solid tumors now ongoing. To manage these patients, it does take a village. So as you can understand, there's issues of coordination, apheresis, transplant unit, to be in an appropriate area of the hospital and to coordinate intensive care unit, neurology consultants, emergency room in case a patient is outside the hospital, needs to come in very quickly. All of these come into one place and work in collaboration with the cell therapy program, and that's done in other centers as well, but this is the team that we've put together here. This is just the last slide to show you what things we already have available here at Columbia. We have access to both commercially available products. They're not yet available for myeloma, but Kymriah and Yescarta that got very good press, but also a little bit of bad press because of the price tag that was given on them. But they're used and very useful in leukemia and lymphoma. These are some of the trials we have for EBV positive lymphomas. Very soon we will be opening a trial with that specific card that I've shown you for relapsed or refractory myeloma, and we have a couple of other trials in planning. So this was my last slide, and I want to thank you very much for listening.
Thank you.